organisms, even the largest, begin life from a single cell. The cell then grows by dividing into two, wherein each parent cell forms two daughter cells, which mature and further divide to form new cells. The process of cell division is vital to all living organisms. During this process, cell growth takes place, followed by DNA replication and cell division. However, the process must take place in a coordinated manner, so that each cell divides correctly and the genomes in the progeny cells remain intact. The series of events by which a cell duplicates its genome, synthesizes its constituents, and eventually divides to form two daughter cells is known as the cell cycle. Although the process of cell growth is a continuous one, DNA synthesis takes place only during one particular stage in the cell cycle. After DNA synthesis, the replicated chromosomes or the DNA are distributed to the daughter nuclei through a complex series of genetically controlled events during cell division. The duration of a cell cycle varies from one organism to another. A typical eukaryotic cell, like those in human beings, divides once in about every 24 hours, whereas in yeast, division takes place in about 90 minutes. Also, the cell cycle is divided into two basic phases, the interphase and the M phase or the mitosis phase. The interphase is the resting phase during which the cell prepares to divide by undergoing cell growth and DNA replication in an orderly manner. The interphase is subdivided into three phases: the G1 phase or gap 1, S phase or synthesis phase, and G2 phase or gap 2. During the G1 phase or the first growth phase, normal cell functions occur. The cell continues to grow and remains metabolically active, but the DNA doesn't replicate. In the S phase or synthesis phase, the DNA synthesizes or replicates, and the amount of DNA in the cell doubles without any increase in the number of chromosomes. Also, the centriole begins to duplicate in the cytoplasm. Finally, in the G2 phase or gap 2, the cell prepares for mitosis and cell division by synthesizing proteins while the cell continues to grow. Now, the next phase of the cell cycle is the M phase, where actual cell division or mitosis takes place. The M phase begins with nuclear division, which is the division of daughter chromosomes, known as mitosis or karyokinesis. It ends with the division of the cytoplasm, which is known as cytokinesis. Did you know that in the 24-hour average cell cycle of a human cell, proper cell division lasts for only about an hour, while more than 95% of the cell cycle duration is the interphase? In adult animals, for example in humans, some cells, like those in the heart, do not appear to display cell division. Animals also have many cells that divide occasionally, only to replace cells lost due to cell death or injury. Such cells that do not display further cell division exit the G1 phase and enter an inactive stage called the G0 phase or the quiescent stage. Cells in this stage remain metabolically active but proliferate only when the need arises. Moreover, in animal cells, mitotic cell division occurs only in diploid somatic cells, whereas in plants, it is seen in both haploid and diploid cells. In living organisms, the cell cycle is a series of events that leads to cell duplication and division, resulting in the growth of organisms. Mitosis or karyokinesis, along with cytokinesis, defines the M phase or the mitosis phase of the cell cycle. During mitosis, parent cell divides into two daughter cells, which are not only identical to each other, but also to the parent cell. Mitosis is also known as equational division, as both the parent and the daughter cells have the same number of chromosomes. Mitosis is a continuous process. However, for convenience, it is divided into 4 stages of nuclear division, namely prophase, metaphase, anaphase, and telophase. Prophase is the first phase of mitosis and follows the S and the G2 phases of interphase. During the S phase, the DNA synthesizes or replicates, and in the G2 phase, the cell continues to grow, but the DNA molecules formed are intertwined and not distinct. However, during prophase, chromatin condensation or the untangling of the chromosomal material takes place, and the centriole that had duplicated during the S phase starts to move towards the opposite poles of the cell. Certain characteristic events mark the completion of prophase. Compact mitotic chromosomes are formed due to the condensation of chromosomal material, and each chromosome is made up of 2 chromatids attached together at the centromere. Moreover, the centrioles that had moved to the opposite ends of the cell initiate the formation of the mitotic spindles with the help of the microtubules and the proteinaceous constituents of the cell cytoplasm. At the end of prophase, the nucleolus, endoplasmic reticulum, Golgi complexes, and the nuclear envelope are not visible when viewed under a microscope. Prophase is followed by metaphase, the onset of which is marked by the disintegration of the nuclear membrane, resulting in the scattering of the chromosomes in the cytoplasm. During this stage, the chromosomes can be clearly seen and studied under the microscope as their condensation is complete. Moreover, each chromosome is made up of 2 sister chromatids bound together by a centromere. Each centromere has a small disc-shaped structure on its surface called the kinetochore. Spindle fibers attach to the kinetochores of the chromosomes, and the chromosomes move towards the spindle equator. The plane of alignment of the chromosomes during this phase is known as the metaphase plate. Metaphase is followed by anaphase. At the start of this stage, the centromere of each chromosome splits simultaneously and the chromatids separate. These daughter chromatids are the chromosomes of the future daughter nuclei. The chromatids then slowly move towards opposite poles. The centromeres of the chromatids face the pole and are at the leading edge, while their arms trail behind. The final stage of mitosis is telophase. This stage is characterized by the chromosomes clustering at opposite spindle poles. Then the chromosomes decondense or lose their individuality as discrete elements, and a mass of chromatin material tends to collect at the two poles. Moreover, a nuclear envelope is formed around each chromosome cluster, and the nucleolus, Golgi complex, and endoplasmic reticulum reappear at both poles. Thus, mitosis is divided into 4 stages, at the end of which a cell segregates its chromosomes into 2 identical sets in 2 daughter nuclei. Mitosis, or karyokinesis, along with cytokinesis, defines the M phase or the mitosis phase of the cell cycle. Mitosis results in the segregation of duplicated chromosomes into 2 daughter nuclei, and is followed by the division of cytoplasm, that is cytokinesis, to produce 2 daughter cells. However, the process of cytokinesis